Well, welcome everyone. Good afternoon or good morning to you all and to the session of Health Data Research UK's One Institute event, which is focusing on the link between molecular science and health records. So you have two chairs for this session. Myself, I'm John Dinesh. I'm at Health Data Research UK Cambridge across the Sanger Institute and the University of Cambridge. And I'll allow Adam Butterworth, my co-chair, to introduce himself.
Hi everyone, I'm Adam Butterworth, also part of the Understanding Causes of Disease team, also based at HDR UK Cambridge.
We have a stellar group of individuals who will be contributing to this session, and we'll introduce them each as the talks come up or the mini presentations come up. Perhaps first to set the stage for the talk with some housekeeping. So there'll be opportunities for two question and answer sessions, one after the first set of talks, and then one at the very end that focuses on the trio of talks on work in consortia of consented population cohorts. Please do use your Q&A function rather than the chat function as you submit your questions. And just let everyone know that the session is being recorded. So just to put this all in context, the understanding causes of disease is one of Health Data Research UK's priority areas. And the underlying theme is linking molecular science to electronic health records at scale. And there are three major programs that illustrate the opportunities and challenges. And each of these programs will be exemplified by science that has arisen very recently in response to the international COVID outbreak and pandemic. So we'll be hearing about genomics and other multi-omics approaches. We'll be hearing about population patient cohorts, and we'll be hearing about health informatics. So to get this going, I'd like to introduce our first speaker, Ewan Harrison. So Ewan is a Health Data Research UK Fellow, and he has in recent months been catapulted into a very important leadership position as a scientific coordinator manager of the COG UK program. And I'll let Ewan explain that to you. Ewan, welcome.
Hi, so yeah, I'm the project manager for the COVID Genomics UK Consortium, known as COG UK. Could you go on to the next slide, please? So the COG UK is a partnership of 15 regional sequencing sites based mainly at UK academic institutions, 2 in Scotland, 1 in Northern Ireland, Public Health Wales and University of Cardiff, and then more locations across England, and also includes the Wellcome Sanger Institute, which acts as a large-scale centralized sequencing hub. So if you can move on to the next slide, the COG UK Consortium came out of a meeting in early March, which discussed the need for a piece of national level infrastructure to sequence COVID genomes, and it works broadly by channeling samples. You can see here on the left of this operational structure, samples from NHS laboratories across the country, either into one of the regional sequencing sites in proximity to them, but also for sites that don't have a sequencing hub close by, into the Sanger Institute. We also receive samples from the UK Lighthouse network. So these are the 3 large labs that are located in England and Scotland that are processing the samples at scale. So this is hundreds of thousands of samples, and the Sanger receives bulk transfers of samples from these for sequencing. All the sequencing data then flows into the MRC Climb server, and we also extract data via the 4 UK public health agencies, and this is also transferred into the Climb server, which is operated out of the University of Birmingham and Cardiff. Making use of this centralized data resource, we have a number of analysis teams who are analyzing COVID genomes from across the UK and mapping that into international data as well. We then feed back data via the public health agencies into government, and we're also in the longer term building ability to feed back to NHS laboratories. We go on to the next slide, please. In terms of sequencing, for those of you who don't know, the SARS-CoV genome is about approximately 30 kilobase in length. And the COG UK takes a sequencing technology agnostic approach. So people can use any kind of sequencing technology that they have access to, although most people use some sequencing technology that came out of approaches that came out of the Arctic Network, which was a Wellcome Consortium grant that existed beforehand. We have an open access data policy, so we release all our data into GISAID, but we also make it completely open access in the European Nucleotide Archive, and we encourage people internationally to do the same. We also have our protocols in online as well, and the idea being that the sequencing technology is only powerful if internationally we're all generating sequencing data, and then we can make use of it collectively. Just go on to the next slide, please. So what's COG UK's major focus? Well, we're interested in introductions and spread. We're also interested in non-chromosomal transmission of COVID, and we've been doing work on that. We're tracking variation in diagnostic primer target sites, and also now we're starting to align with studies that are testing out new drug strategies, and also we obviously have interest in variation in potential vaccine antigens. We're also doing transmission tracking at scale, both nationally and locally, and we're involved in tracking outbreaks and seeing what the effects of interventions are. We have a very strong core group of analysis team who are involved in evolutionary biology. So we have a very strong evolutionary focus as well. And we're also starting to track the emergence of viral variants. We're also, particularly from a health data UK approach, really interested in linking clinical databases into as I mentioned previously, into to viral data. And that's a piece of really huge ongoing work at the moment. We'll just go on to the next slide, please. So in terms of our ongoing analysis, we've recently published information about the early spread of COVID into the UK, and the fact that there were probably at least 1,500 independent introductions, and that most of those introductions likely came from Spain, France, and to a lesser extent from Italy. We've been monitoring the emergence of dominant lineages in the UK, estimating R in London. We've also been doing tracking transmission in hospitals and care homes, and COG UK members have been publishing papers on this. We've also been interested in the effect of these spike protein mutations, and we're now starting to link health record data to the viral data to see what the effects of these mutations are. Are they associated with worse outcomes, etc.? Let's move on to the next slide, please. And finally, what's the future? So as I mentioned, there's a major piece of work now going on in terms of linking the viral data to hosts. We're working in partnership with a genomic study, which you're going to hear about a bit later on. This is to the host genome sequencing, so we're ensuring that we have coverage of people with viral sequences from host data, and we're also working very hard to ensure linkage with a broad range of clinical records. We're working internationally in terms of analysis, but also knowledge exchange, and we attend regular international meetings. We're also hoping that this becomes a national part of the national infrastructure going forward. Obviously, there's potentially a second wave, but this kind of approach is also really amenable to future pandemics, particularly flu and other respiratory pathogens. And a final piece of work that's really going on is to how can we report effectively information back into the NHS. And that's the summary of Health Data Research UK. I'd just like to go to the last slide, please, just to really indicate this is a huge consortium effort. I'm just a spokesman for it. There's over 400 people who've been involved in this effort, so it really is a really huge national effort. And you know, without any one of these individuals, there's huge parts of it that just wouldn't work. So I'd like to thank all my co-consortium members.
Team Science writ large, thank you, Ewan, from NASN to Zarebski on your list of co-authors. So thank you very much, and Ewan, perhaps understating in some ways the international prominence of this effort. I believe that more than half of the globally published coronavirus genomes, more than half have come from this particular initiative. Ewan has alluded to its connections with host susceptibility and host genomics, which is a nice segue into the talk for our next speaker. So Professor Sir Mark Caulfield Amongst many other roles, Mark is the chief scientist, has been chief scientist for Genomics England, Queen Mary University in London. And today Mark's going to tell us about this exciting new initiative, which has linkages with what you just discussed, called UK. Mark, you're welcome.
Thank you very much, John, and thank you all for having me here. I'm going to talk about whole genome sequencing in COVID-19. And this is a partnership between Genomic, a study established in Edinburgh University by Dr. Kenny Bailey, and Genomics England, along with the partners that you see at the bottom, which include all the public health agencies, the NHS, and of course, Health Data Research UK. So if we go to the next slide. The importance of host response is underscored by the variability of individuals' trajectory with this disease. And we've seen both in COVID-19 and in previous coronavirus epidemics, very variable response on exposure to the virus. Some people end with needing support in intensive care, and some of those sadly perish. As many as 56% of those that make it to ITU, based on ISARIC 4C data, will sadly die. So understanding the variability in this response and why some people are very, very mildly affected or even asymptomatic but exposed to the virus, and understanding why for others there's a very severe outcome is really, really important. This is especially underscored by the outcomes that are much more severe in individuals of our BAME communities across the UK who are traditionally underrepresented in research. So in partnership with the University of Edinburgh, Genomics England and Genomic designed a programme to look at up to 20,000 severely affected individuals with COVID-19. And the idea is that we will compare those to 15,000 mildly affected individuals or asymptomatic individuals, largely detected from either viral screening or alternatively from seroprevalence studies. And we're embarked on doing that right now. So the severe versus mild is essentially an extreme comparison. We will enrich that by COVID positive cases or COVID tested cases from the 100,000 Genomes Project, and also where post follow-up with primary care, hospital episodes, other secondary care data sets and registries, including the ITU national audit, which we are also collecting. We have today enrolled over 2,700 people in the program, and that's continuing to increase each day, and we reach across over 200 ITUs across the United Kingdom. That's allowing us to enroll at speed and pace, having started the study just a couple of weeks ago. We are also linking, as Ewan mentioned, with COG UK's viral program, and that means that we'll be able to look with COG UK at within virus, within host, and a combination of the two for the first time. This program can take heart from a recent preprint on Medic's RIV, if you want to look it up, of a Spanish and Italian study of severely affected individuals versus population controls, where two loci in 1,800 cases versus 2,200 controls have shown genome-wide significance. And those loci track to a transporter, a solute carrier transporter that affects the ACE2 protein, which is the method of entry of the virus inside the cell, as you know. And then the other area is the ABO blood group, particularly focused on blood type A, and in adjacency to the von Willebrand-Proctor gene locus. So these are two loci genome-wide significance in what's essentially less than a tenth of the study size that we propose to undertake. Now, to be open and candid, in the first wave, I expect we'll reach about 5,000 to 6,000 people, but we are now ready and enrolling for a second wave, should that come. So we will undertake our first analysis, we hope, towards the end of July, and we are whole genome sequencing now. So we've created a new research environment to host this, which is provided by LifeBit, which is much more user-friendly than our existing Genomics England data center. And that will be in the Amazon Web Services cloud. That will therefore allow connectivity to other programs across the world. So we formed links to other programs in Canada, Qatar, France, and we have also joined the International Consortium in COVID Genomics. And so what we hope to do with the research community worldwide through our Genomics England Clinical Interpretation Partnership and with a pre-competitive industry consortium is to make insights into new biology that may tell us a number of things. Firstly, this may identify risk factors that will allow us to encourage certain people to shield more rapidly to respond to a new health emergency. The second is to identify potential repurposing opportunities for therapies that could be trialed. The third thing is we're trying to enroll as many people involved in trials as possible with the goal of being able to look at the genotype response correlation between the drug and the individual using the whole genome, and also to be able to look at signals of harm, if there are any, for the therapies under test. So we then, in addition to that, we may be able to look at comorbidities because we'll have extensive healthcare data. So I want you to imagine that we've collected as much health data on anybody as we can from public sources, and including rich data from the ITU National Audit Project, and that this data will be available for UK and international researchers worldwide. That's you. And we hope that you will use that to make new insights into this terrible disease so that we can advance the care of patients in a secondary or tertiary wave. And this study is made possible by funding from the Department of Health and Social Care, also from the UKRI, and also from Genomics England. So I'd like to take this opportunity to thank you, John and HDR UK, for having me here today. And if I could just go to my last slide, please. I'd like to acknowledge the myriad of people, a bit as Ewan did, but not with names on the slide at the moment, who are and who will take part in that. And I very much hope you will join in the research endeavour to bring these data to life for our patients. Thank you very much.
Thank you very much, Mark. We have a few moments now for questions, and I'll kick off for a question which is, I think, directed both at Mark and at Ewan, which is about sampling strategy in each of the initiatives we've heard about, Genomics and COG UK. So Tony Brooks asked the question, to what extent have these initiatives focused on extremes, and then parenthetically, young, healthy people that die, old, ill, who survive, most divergent virus strains and their clinical impact, which is often where clues can be found. So it's about using the extremes strategy. To what extent is being used? Perhaps could you tackle that first, Mark?
Yes, John. So great question, Tony. The program does focus on trying to enroll across the whole community, but particularly in the younger groups. So if I share with you that today we've enrolled 274 people below their 40th birthday and 709 below their 50th birthday, you can see out of 2,700 that are enrolled today, you can see that we are trying to capture as many of the individuals who will be in the younger age group. In the first decade of life, we have just over 10 children, and you will have noticed that there's been a particular syndrome that mimics Kawasaki syndrome in some of the children. In at least one of the patients tested, a rare variant, and that's the virtue of whole genome sequencing, has been found that's paved the way for an immunotherapy in the NHS that has allowed that young person to recover. In the very young, you are correct to suggest it's possible that a genomic architecture may be more prominent, but obviously we will investigate that as a component of what we do here.
Thanks, Mark. Ewan, quick comment about sampling strategies being used in COG UK. There are multiple.
Yeah, so we have a sampling strategy that aims to capture a random snapshot in terms of surveillance across the UK, and we do that via our sequencing sites. We're also enriched for samples from ICU as part of our study to link with the genomic study. But we also have a number of other sub-studies that are embedded in that target various different populations. And we're also very keen to link up with things like the ONS surveys, which will capture really the kind of the full range of people in the community as well. So that's the people who have asymptomatic disease.
Thanks very much. I think in the interest of time, we'll move on to the second limb of this session, which is about consented cohorts and multiomic data, and relates to Alex Forrest Hayes' question, which I think the speakers will collectively address, which is about samples to explore other omic streams other than genomics. So I'll hand over to co-chair, who now turns into presenter, Adam Butterworth.
Thanks, John. So I'm speaking today in my role as the lead of the HDR UK National Implementation Project for Multiomics. And I'll tell you a bit about what that is and what it plans to do. And it's great to be joined by Emily Jefferson and Arun Hingarani, who are work package leads in this, this project, who can talk about different facets of the work that's going on and how we've tried to repurpose to some extent this nascent national project to deal with the COVID pandemic. The project started formally in April of this year, so just about the time when a lot of attention was being diverted to other projects. And so we'll hopefully give you a flavor of what's happening and what will happen in relation to COVID from this effort. Next slide, please. So first of all, I thought it might be useful just to address the question we've heard already from various talks in this session and in earlier sessions about the value of collecting COVID patients and doing various kinds of research on them, including genomic and other types of research. Why is it informative? Why is it useful to take existing population cohorts that we have around the UK and try to use them to study COVID-19? Well, clearly it's efficient in some ways because there's extensive data been recorded from many participants around the UK in existing population studies. We've heard about UK Biobank already, but of course the UK has many population sampled cohorts around its breadth. This avoids us having to go out and do new measurements. There are also challenges with doing measurements during the acute phase of disease. So perhaps for genomics, as we just heard, that might be fine. An individual's genome doesn't change due to having had COVID-19. But of course, factors like proteins or metabolites, other things we can measure in the blood may well be influenced by the onset of disease. That may be very useful for telling us clinically about what's going on with the disease course, but it may be less useful for telling us things about risk factors for disease or risk factors for disease severity. In a similar way, by having a prospective design, and by that I mean by taking information that we measured before the COVID pandemic hit the UK, we can avoid the idea of reverse causation, the problem of measurements being influenced, whether that's our recollection, our memory of events and risk-taking behaviors, or in this case, whether it's measures of blood-based molecular factors. We can avoid this problem of reverse causation. One of the other big challenges that we have when we try to study a disease like COVID is the potential for bias. And by that, we mean the potential for biases to arise, for spurious associations to be found when we look at selected groups of participants. And when we try to study just certain types of patients, COVID patients or hospitalized COVID patients or severe COVID patients, we have the problem of trying to find an appropriately matched control group that avoids some of this distortion that can arise. By taking a prospective cohort approach and by essentially starting with a sampling frame designed some years ago and then seeing which of those participants go on to develop COVID, which ones have COVID more severely, which ones have different outcomes post-COVID, we might be able to minimize some of those biases. So we think there's great value in using existing population cohorts to try and tell us something about COVID disease. Next slide, please. And just to say, the bullet point at the bottom now is just saying we really think there's valuable insights and that this is really complementary to patient collections and the valuable insights that will be gained from those using an existing population cohorts. Thank you. On to the next slide, please. So what is the HDR UK Multi-Omics Consortium? Well, it's a collection of population cohorts from around the UK. It involves many investigators from many different sites that you see here on the map on the left. And pleased to see that many of the investigators centrally involved in that have joined the breakout session today. And the distinguishing features of these cohorts is that they all have genetic data as a backbone. They all have linkages to electronic health records, and we'll hear more about that later in the session. And they all have some aspect of multi-omics data, whether that's RNA transcriptomics, whether it's proteomics, metabolomics, some other kinds of multi-omic data, sometimes imaging, for example. Not all necessarily the same across all the cohorts, but useful different assays that can be harnessed for study of disease. Next slide, please. We hope that by integrating this information, the genomic data, the multi-omic data, the health linkages, we can identify molecular factors associated with things like risk of COVID-19, severity, complications, etc. Using the genetic data particularly, we can try to put this in a causal framework, as Arun will unpack in the following talk. This will inform our understanding of the causal role of different biological pathways in COVID-19 and outcomes. It will hopefully help us build and test predictive models of the sort that Mark Caulfield was talking about. How do we identify the most vulnerable people, not only on the basis of clinical or physical characteristics, but also biological characteristics? And we hope that it will also help us inform therapeutic strategies that we may use to help treat the disease. Next slide, please. My final slide is really just to highlight some of the challenges and how we're going about solving those. Firstly, statistical power. We heard from Mark Caulfield about the plan to sequence 20,000 genomes, but perhaps in the first wave, we'll only have maybe 5,000 participants who can be recruited. How can we make sure that we have enough people from these prospective cohorts who had COVID to enable us to have power? Well, there are various solutions to this, such as scaling up and broadening the multi-omics consortium, and we're inviting more cohorts to take part, as well as collaborating nationally and indeed internationally. And some of the cohorts are already joining the COVID-19 Host Genetics International Initiative to share data worldwide. How about the availability of relevant outcomes and COVID-linked data? Well, we've already heard this morning about some of the challenges to that, but what are some of the potential solutions? Well, we're looking to share our initial learnings from individual cohorts across the consortium and work out how we can work together to streamline the effort. And particularly important in helping to support the data providers. How can we make it most easy for the people linking these records to the individuals? How can we make things easier from their end and provide the necessary support that makes it a possibility? We've already touched on the methodological biases, and we're working very closely with methodologists to try and understand them and minimize them and think carefully about our phenotype definitions and the comparisons we do. And then finally, as Emily Jefferson will talk more about, how about the computational infrastructure? How can we bring these data together? How can we analyze them in a efficient and compatible way? So I hope that's given you a flavor of the multi-omics consortium and what we're attempting to do there. I'll finish there and move on to the next talk in the session, which I think is Professor Arun Hingorani, who's a Professor of Genetic Epidemiology from University College London. He's also a director of the UCL Institute of Cardiovascular Science, and he's going to talk to us about the implication of multi-omic cohorts for medicines development. Arun, over to you.
Thank you, Adam. Thanks very much. Good morning, everyone. Could I have the first slide, please? So I think we're all well aware of the various strands of activity that are underway to try and control the spread and transmission of the virus and reduce the adverse consequences, from public health measures on the left, of which we're all participating at the moment, to attempts to develop a vaccine to provide protection, but also now to try and interrogate the possibility of developing medicines that might protect susceptible individuals from viral infection, or if they are infected, to reduce the adverse consequences. So viral infection with coronavirus and all other viruses involves an interaction between viral proteins and host proteins at the level of viral entry into the susceptible cell type, viral replication, and also host proteins, which might determine maladaptive responses to the viral infection. And in the case of coronavirus, COVID-19, it would appear that some of the adverse consequences of the infection arise due to a maladaptive or overexuberant inflammatory or procoagulant response in the host. Now, given that most small molecule and biotherapeutic drugs act on proteins, understanding which viral proteins and which host proteins are involved in pathogenesis is likely to be important. Now, there are several approaches aimed at identifying drugs that influence the viral entry and replication cycle by targeting viral proteins. But the approaches that Adam has described, and Mark Caulfield as well, the genomic approaches, provide potential to identify proteins in the host that might be amenable to targeting to block processes by which the virus enters or replicates, or to damp down or ameliorate or reduce maladaptive host responses. Next slide, please. So the principle that might be applied is one that's becoming widely used, and that's to use variants in a gene encoding a protein that might be involved in viral pathogenesis, a host protein, and use those variants in a gene with measures of the proteome to try and predict or anticipate what the effects of a small molecule or large molecule drug targeting the same protein might be on the response to the virus and perhaps the maladaptive host response as well. So this approach has been termed use of Mendelian randomization approaches for drug target validation. Next slide, please. So what I wanted to do here, and this is with permission of Mark Pietzner, Nick Wareham, and Claudia Langenberg from HDR UK Cambridge and the MRC Finland cohort, is an attempt to try and identify those proteins in the host that might be involved in viral pathogenesis. And what Mike and colleagues did here is curated a whole range of proteins on the left-hand side shown here that relate to viral entry, that have been identified in cellular studies from other groups as potentially being involved in the host protein, viral protein interaction as part of the viral pathogenesis, as well as proteins associated with adverse outcome from COVID-19, using proteomic approaches and also by scrutiny of clinical biomarkers of outcome from electronic health record studies. Now, a number of these proteins are now assayable using large-scale proteomics platforms. In this case, it's the one from SomaScan or SomaLogic. Here, about 5,000 proteins have been measured among 10,000 participants in the Finland study, and about 179 candidate proteins overlapped with those that are identified as potential candidates in the host involved in pathogenesis of COVID-19. Now, an advantage of cohort studies is that many of them already have genotype data, so it's been possible to identify genetic variants acting in cis that affect the circulating concentration of some of these candidate proteins. So now, coupled with emerging GWAS data from case control collections that Mark Caulfield described, or cohort collections that Adam Butterworth described, it may be possible now to try and identify, using Mendelian randomization approaches, it may be possible to model the identification of proteins that are druggable targets or drug targets that might modify susceptibility to viral infection, to replication, or the adverse host response to COVID-19. So this is an illustrative example of how multi-omics data applied in population cohort studies might be used to gain insights into therapeutic targets for the management of COVID-19. And I think sort of serves as a sort of proof of concept example to showcase how research might be taken forward. Thanks, Alan.
Fantastic. Thanks, Rune. That's wonderfully tantalizing data to give us examples of the kind of outputs that one might get from this kind of work. Excellent. So next, the last speaker in this breakout session is Professor Emily Jefferson from Dundee. She's Professor of Health Data Science and Director of the Health Informatics Centre Services. And she's going to talk to us about the infrastructure aspects of multi-cohort analyses.
Everyone, so I'm just going to talk for a couple of minutes on what we're doing around safe havens and enhancing them in order to enable access to this fantastic data. Next slide, please. So this is the beautiful diagram that we put in for the grant proposal, looking at the various different work packages. And so this particular bit that I'm working on is around work package 1, and it's looking at the informatics infrastructure. And what we're doing at the moment is doing the scoping work to understand what we need to do to enhance these safe havens so that they can manage this sort of data. Next slide, please. So this is the way I see it. I originally started in the world of bioinformatics, where all of the data was kind of relatively easily accessible. There weren't any issues with patient confidentiality, and there wasn't any issues with sharing data and sharing code and things. I then went away, worked in industry for a while, and then came back and started working in health data, and found that the worlds were very, very different in terms of how easy it is to share code, to share data, and in order to kind of build those infrastructures. And so I think because kind of bioinformatics has this sort of openly accessible environment and a community around it, I would say that they've managed to kind of move forward very quickly in building collaborative tools. Whereas within health informatics, because it's been more restricted, the community to share resources that are dealing with public patient level information is less far along on its journey. So I kind of see this as bridging a gap between two worlds of bringing the patient level clinical data to the world of the kind of bioinformatics data, and trying to find a bridge which will help us to bring. Next slide, please. So, trusted research environments, also known as safe havens, have been around for many decades as a way of actually accessing data in a secure way, where the data can't leave the environment, but people can go in and they can use sort of stats analysis within that environment. So it's been great for kind of epidemiological studies, where that's all you've really needed in order to analyze the data. But when you're dealing with other data types, kind of big data, and you're needing programming access, these environments basically aren't set up in order to do that. They've been set up with the idea of epidemiological studies. And so there's a range of us kind of working in this field to try and create what I describe as kind of next generation safe havens. And these are environments where they're still locked down. You still can't sort of have free access to the internet, but you can still provide the sorts of tools that researchers need in order to carry out their jobs. So what we're trying to do is look at secure ways of researchers being able to kind of install the software and the pipelines that they want in an easily maintainable and automated way. But we're also looking at the kind of the hardware underneath. These safe havens are kind of providing GPUs rather than just CPUs, and also we're looking at how do you go about exporting algorithms that might be developed. So with stats analysis, it's quite easy to do disclosure control, because you can look at a graph which comes out as your final result. But with kind of AI development, you're actually wanting to export an algorithm, and sometimes algorithms can be trained in such a way that they actually inadvertently store the row level information if they're really deeply trained. And so we need to make sure that when people are exporting algorithms and exporting software from the environments, that's a secure way of doing things. So the bit of work that we're doing at the moment is very much in collaboration with Sanzi, with Chris leading the work package with me. And what we're doing is a kind of a landscape review at the moment, looking at what different options are out there, and building on that to create what we hope will be a next generation safe haven or trusted research environment. Thank you very much. That's me.
Thanks, Emily. Fantastic to hear about the work that's going on, and how we're going to be able to actually operationalize all these wonderful analyses that people want to do. We've talked about this across the UK, but clearly COVID-19 is a global problem. It'd be interesting, I think, both from the analytical infrastructure perspective, but also from the more applied perspective, to think about what's the potential to scale this more internationally, do you think? Emily, can I start with you on that?
So I think the idea with TREs and the processes that we're trying to do is that they should be safe environments for collaboration in a federated way. So whether that's across different groups within the UK or whether that's different groups internationally, the whole idea is that we're kind of putting in those processes and those secure ways of managing data to enable enable access across the world. So the trusted research environments can be are virtualized, and so they can be accessed from different places. But there's a sort of set of principles around how we manage this sensitive data. And because those principles are there, it should mean that others from across the globe can access the data, but equally that we can work with those people as well. So the whole infrastructure is essentially designed to scale both on a UK-wide level, but also across the world.
Thanks, Emily. Arun, what do you think about the international perspective here?
Yeah, thanks, Adam. Yeah, as you've mentioned previously, there are a number of initiatives emerging internationally, such as the COVID-19 Host Genetics Consortium and many others. So certainly the availability of data won't be lacking. I think the challenges are around processes for analysing the data jointly, either using federated approaches or trusted research environments. And the other challenge also is harmonising outcome data, the phenotyping of the disease endpoint, for example. And it's sort of gratifying that there isn't another HDR UK national implementation project in phenomics, which is helping to address some of those issues. And I think we'll also partner and work internationally to ensure that these disease definitions and subsidiary outcomes or complications of COVID infection are sort of coded in a standardized way that's comparable across data sets.
And I guess that's particularly important with COVID, whereas we've heard already during the day the numerous different ways of collating information about COVID, symptoms, tests, etc. That will be particularly important. We've got a question on the Q&A coming from Georgie Humphreys, who asks, how do we build credit and incentives to encourage data contributors to actually share? Arun, do you want to tackle that one?
Yeah, so I think it's a little bit about pre-existing cultures. So certainly in the genomic world, I think we've gone through a decade or more of genome-wide association studies with very large multi-author papers where systems have been put in place so that the sort of younger members of teams who are driving the work forward, doing the heavy lifting, are recognized and have visibility, but also that the sort of key participation of all the data, of all the people involved in producing the data and bringing it together are also recognized. So I think, there are many new models of research emerging. COVID-19 has sort of compressed the way we do research in terms of timeframes and is invoking new models. So it's a moving field, but it's something that we do very definitely need to be aware of. Research is done by individuals, and individuals need recognition and credit for the work that they do.
Emily, did you want to come in there?
Yeah, I think I think there's two different things here in terms of a carrot and a stick. So in terms of the stick, I think it would be really great if funders supported this sort of initiative in terms of the data sharing. So obviously, some of the principles that have come out is very much around if this is data that is created, then there's an encouragement to share data which is collected. But I think perhaps the funders could be slightly more encouraging with a stick in terms of sort of having to share some of the data and kind of work with these wider initiatives. In terms of the carrot, I think the very nature of for one particular initiative collecting data, then being able to do meta-analysis across lots of different data sets if they make their data available, I think produces it gives a nice carrot basically. So what we're wanting to do with a lot of the data across the UK is mean that the data becomes available not just for kind of finding the data that is out there, but also that you can query the data sets in an automated way and a secure way across those different and so only in making your own data available for querying can you then have access to query across those data sets. So there's a nice carrot.
I think the COVID-19 emergency has really given a great opportunity to progress this data sharing and for people to really collaborate and work together that has been seen in some areas before, as Arun was saying, but maybe hasn't always been seen. And I think this is something that we need to try to harness for a post-COVID world, as well as the infrastructures and the linkages that we've talked about. The data sharing collaborative nature is one that we really need to continue to push. We've got a question coming from Ros Walker from HDR UK, who says, can you predict the most likely early disease insights for COVID-19 and more broadly that might arise from this great team effort? Maybe I'll start with that one whilst Arun and Emily are thinking about that one. I think Arun's already shown some nice early data from the Fenlon cohort that's starting to show how some of the proteins that we've already measured in some of these cohorts might be turn out to be causally related to COVID-19. I think in the first instance, what we're going to see is probably reasonably large sets of molecular risk factors that seem to associate with different COVID outcomes. So infected versus non-infected, severe versus mild, death versus recovery. I think the question then is really how we progress those from initial association analyses to something that we can then use to make a difference. So is that predictive tools? Is it the causal insights that Arun talked about from Mendelian randomization? Is it taking it into laboratory studies to try and unpick the functional mechanisms of why these particular markers might be related to COVID? Arun, do you have thoughts there?
Yes, I'd agree. I think we will learn a lot about the host response to COVID from genomic and molecular studies in tandem in human data sets, in consented cohorts, and in the case control collections that Mark's described. And then the challenge will be to try and pinpoint whether any of the mechanisms that are unveiled are actually targetable by therapeutics, and whether they're targetable by drugs that already exist and might be repurposed. So that would be a fantastic goal if we could achieve it. But of course, the data will speak, and we need to do the work to see whether there are some therapeutics that can be brought into the frame early on to modify the course of the disease or prevent severe forms of the infection.
We had a question from David Seymour, who asked about how well represented are different ethnic groups within the multi-omics cohorts, and could international collaborations help fill any gaps? Almost certainly, I think, is the answer to that. So definitely international collaboration will help fill the gaps. We've already touched on in this session different response, perhaps by ethnicity to COVID, and that's one of the areas that is obviously a very important topic right now. Is there any biological underpinning for differences seen amongst different groups of people, particularly different ancestries? In the UK cohorts, there are different cohorts that sample from different areas. So for example, some of the expansion that we've been doing, bringing in cohorts, for example, in Bradford and Leicester and in London, will help to build our ethnic profile beyond European ancestry participants, but undoubtedly, international collaboration will help with that. I'm conscious of the time. I think we have one more minute in this session. And so maybe we can just quickly address a comment or a question from Colin Wilkinson, who says, how do you ensure the people whose data and samples you use are involved in your work? This is going to help public trust. A key aspect of the Multi-Omics Consortium from HDR UK and from, in fact, all of the HDR UK programs is the importance of patient and public involvement. I think all of the programs have embedded within that so-called PPI&E involving patients and participants right from the onset. We have a panel, PPI&E panel, that is part of the Understanding Causes of Disease theme within HDR UK that inputs to all of the programs that run in this area, including the multi-omics cohorts. And of course, many of the cohorts, if not all of the cohorts in this consortium, also have their own panels who are involved in the research as well. So I think that's an absolutely critical question, critical point, and something that we and HDR UK are very keen to address, particularly around the public trust issue. So we've reached 12:35. Excellent session. There are more questions, so great to see them coming in and great to see the interest, the attendance from everybody. Thank you to all the speakers for fantastic talks. I hope it's really given people a great overview of some of the exciting research that's going on in the UK health data research community, and some of the things that we can look forward to in the coming months, helping to address the COVID pandemic, and how really the community is really pulled together to mobilize and to transfer existing data sources to fight COVID, as well as building fantastic new resources like the COG initiative that we heard about. So thank you to all the panelists and attendees.
Can I just add that we're going to be talking more about trusted research environments in session 7 later on this afternoon, if people are interested.
Excellent, look forward to it. Thanks everyone. Bye-bye.